mientras sea la primera speaker, los demás ahí están.
¿Ya tú eres la jefa?
Sí, 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 bueno, no, están entrando un segundito.
Sí, sí.
We are reaching to the end of the Global Summit on Neurogenetic Diseases. You can. And for this closing session, we have three outstanding neuroscientists, and it's my great pleasure and honor to introduce all of them. The first speaker is Isabel Fariñas, that we all know very well. She's a member PI of the CIBERNED. Also, she's member of the steering committee, scientific steering committee of CIBERNED. But overall, she's a professor of cell biology at the University of Valencia. And she's a director of the Molecular Neurobiology Lab at the Institute of Biotechnology and Biomedicine in Valencia. Her high quality research focuses on the mechanisms that regulate stem cell behavior in normal and diseased brain. And her talk today is immune vigilance of adult neural stem cells. Okay.
Good morning. It's great to be back in person and see you all again here in Salamanca. It's been tough these past years. I would like to talk about what is our major focus in the lab, which is the study of adult neural stem cells and their regulation. And today I would like to focus my talk on how neural stem cells encounters with immunity in the adult brain. So let me show you the context first. We have heard from Julia also about a neurogenic niche in the brain. There is the second one, which is the one that we study in the lab, which is called subependymal zone. And in this niche, we have neural stem cells that, when activated, divide and produce progenitor cells that, after a few divisions, become committed to neuroblasts that exit cell cycle and differentiate into olfactory neurons in the mouse. So these cells can give rise to, these neural stem cells can give rise to neurospheres, and this is an in vitro model that has been extensively used to study adult stem cells in mice to understand the qualities and properties of any adult stem cell. In this niche, these neural stem cells are astroglia related. So they have markers of astrocytes like GFAP and GLAST. And as in other adult niches, we now know that they can be in different states of activation. From quiescent neural stem cells to activated ones. When they become activated, they put in at their membranes the receptor for the mitogen EGF receptor. And we also know now that we have an intermediate state of this, in this activation cycle, which are cells that are molecularly in a quiescent state, but are primed for activation, so they are called primed cells, primed neural stem cells. So in the last few years, Germán and José Manuel in the lab put a lot of effort in devising a methodology to classify the cells of the entire neurogenic niche, or the entire neurogenic lineage, to be able to isolate the different populations, to put them in vitro, and to do functional assays. And this took a lot of effort, but within the population of limb-negative cells, which excludes other cell types in the area that are not neurogenic, they could devise this method based on different markers to prospectively identify and obtain the different cell fractions. And here you can see that we could isolate populations that were potentially prospectively activated, primed, and quiescent NSCs. And we have done a lot of functional studies on these cells, and we have evaluated a lot of inflammation, but that has been published, and therefore I'm not going to talk about that. But I would like to mention, just to mention, that these populations, when we deep sequenced them, we found that they have coherent transcriptomes, and furthermore, if we compare those transcriptomes to transcriptomes generated at the single-cell RNA level, at single cell level, we find also coherence with those transcriptomes. But these cells can be assayed. So what's the problem that I want to tell you about today? We have been, for many years, working on niche biology. We have evaluated how different signals and cell types and elements of this niche regulate this transition between quiescence and activation. And recently, well, a few years ago, we decided to evaluate the role of immunity. And we have been doing work on microglia, which you all know that it's the resident macrophages of the central nervous system. And part of this work has been shown in a poster during these days. But more recently, we decided also to evaluate adaptive immunity. And this is the work of a very, very talented PhD student, Pablo Martí Rodrigo, and this is his thesis work. And we have done this work in collaboration with an immunologist at the University of Valencia, Alberto Yáñez, because we are not immunologists, and both the nervous system and the immune system are highly complex, so we really required an expert in this. So the question that Pablo is trying to answer is if adult neural stem cells are immunologically special. And a more precise question is, can they evade T lymphocyte-mediated killing? And the idea is that these cells are long-lived. They remain, some of them remain for the entire life of the animal. And they have to be preserved because they sustain cell production, not only in the brain, but in many tissues. And as I told you, some of them are quiescent, some of them are activated, and we don't know whether if these cells become infected or they acquire mutations. We don't know whether the immune system is going to attack these cells to eliminate them. So we have to keep these precious cells and at the same time avoid, for example, the effect of mutations that could transform them. So just a quick, I'm going to take you through what happens with a cell generates a neoantigen because it's infected or generates a mutation, and we have been talking these days about abnormal proteins that are related to neurodegenerative diseases, right? So those are neoantigens also. So every nucleated cell in a body exhibits a sampling of its antigens. Self and non-self, so the ones that are characteristically natural of that cell, and those neoantigens that come from the proteolysis of proteins in cytoplasm. So this is sampling, and this is presented at the surface by molecules. A complex is the MHC1 complex. In humans, these molecules are the HLA proteins. And so this is in any cell. These abnormal proteins, these neoantigens, also get to the body fluids, and we have seen many examples, and that's why we evaluate biomarkers in CSF or blood. They get to the blood and to the lymphoid organs where professional antigen-presenting cells also digest these proteins and present them in surface through another type of MHC molecules, which are the MHC class II. Then T lymphocytes, which mediate cellular adaptive immune responses, rearrange the genes that code for the T cell receptors that are also specific, like antibodies. They work like antibodies. They are specific for specific antigens, epitopes. So they circulate to go to the lymphoid organs, and if they recognize this antigen, and if this is a neoantigen, it's not a natural one, they become activated and become cytotoxic CD8 plus T lymphocytes, and I'm going to call them from now on CTLs. Okay? Once they become activated, if they find the same epitope in any cell, they will kill the cell. And that's how this cellular part of the immune system works. But again, let me take you back to the problem. Let me take you back to the context. We are within the brain. And for many years, for many of us, the concept was the brain is immunoprivileged. That's why even in neurodegeneration, we have had a talk on adaptive immunity and Parkinson's disease by David Sulzer. but it's not an issue that has become clear until very recently. And the reason is we thought that the antigens in the brain, proteins in the brain, did not get to the lymphatic system. And now we measure them in blood, but for many years we thought there was no lymphatic system in the brain because there is not. But some years ago, the Kipnis lab described this lymphatic system. So molecules that are present in the CSF do get to lymphatic systems in vessels in the meninges because throughout the perivascular spaces, these proteins can move and can drain to circulation. That means these molecules get to the circulation, to the lymphatic systems, and if they are normal, are presented through MHC2 complexes in professional antigen-presenting cells. But furthermore, more recently, it has been shown by the same lab that there are in the skull, there is a bone marrow with immune hubs where specific T cells are produced that can monitor the brain and can enter the brain. For many years also, we thought that immune cell trafficking was very low in the brain, and it still is lower, much lower than in the periphery, but there are T cells trafficking into the brain, especially if they are activated in these sites. The other question was MHC1, and we heard David Salzer already. Neurons in the adult do not exhibit MHC1 complexes at their membranes, but they can do so during aging or pathology in response to cytokines like interferon gamma. And that's why they can, for instance, put at the membrane epitopes of an abnormal alpha-synuclein, as David showed. So all these things prompted us to consider that maybe in the brain, we have a need to understand what's the relationship between adaptive immune cells, CTLs, and cells that are within the brain. And we started with the adult neural stem cells, which are the focus of our work. Okay, so how about the strategy? If these cells trafficking throughout the CNS is in low numbers, there are several strategies to increase the interaction so they can be studied at the cellular level. And a very cool strategy, I like it very much, is one developed by Judith Agudo, a Spanish scientist now at Harvard, when she was a postdoc in Brian Brown laboratory. And this is the JEDI mouse strain. So what Judith did was to generate the mouse, genetically engineer, so all its T cells have a unique T cell receptor that recognizes an epitope that is in GFP or enhanced GFP, which is this epitope here. So the idea is that if you take cells from the JEDI mouse, and transfer them into a mouse that has cells that express GFP, and at the same time you vaccinate the mice with GFP to activate these lymphocytes, these CTLs are going to kill any cell expressing GFP in the body, okay? They used it to ask the same question that I'm addressing today. What happens if the, GFP is expressed by stem cells in any tissue? And they used LRG5 GFP mice because LRG5 is a marker of many adult stem cells, like the mammary gland, intestine, the skin. And they found that T cells were capable of killing all these stem cells except in resting hair follicles. The hair is cycles, so some hair follicles are resting, some of them are active, and in those resting hair follicles, the CTLs, the JEDI T cells, could not kill the cells. And I'm going just to mention this, they found that these cells that in the resting state do not exhibit MHC1 complexes at their membranes because one of the transactivators of the genes that encode for the subunits of these complexes is not active in the resting stem cells, okay? So Pablo decided to go into our system, and to do so, he started by using a dual reporter mouse line, and in this dual reporter, all cells express TD tomato, but this cDNA can be excised by a Cre recombinase, and then that unleashes the expression of a GFP So we crossed the mice with GFAP-CRE recombinase because, as I told you, neural stem cells in the adult subependymal zone are GFAP positive. So that would label the neural stem cells and the entire progeny, the entire neurogenic lineage. So here you see that when the cells either express the tomato, but if recombination takes place, they lose the red reporter and acquire the GFP expression. And these are different populations in our lineage, and almost 100% of all of them become GFP. Now, the idea was to inject, we can, in this progeny, this F1 progeny, Pablo injected the GDI T cells and vaccinated with lentiviral encoded GFP. And one can see that this is the appearance of the spleens in these animals injected with the GDI CTLs. This is the size of the spleen. All the lymphocytes in the spleen are activated, this marker for activation. We can also detect it, we can isolate them from the subependymal zone, so they have infiltrated our zone and they are activated. And we can also see them by immunofluorescence. We can see that they abandon blood vessels and they get close, especially to cells that are Ki67 positive, which are activated proliferating cells. And these lymphocytes are larger, so they are activated, and then they themselves are Ki67 positive, another sign of activation. So what happens to the populations when these lymphocytes access our niche? So this is the phenotyping of our populations with our cytometry marker panel. And what you see here is that active cells that are proliferating are being lost because they are being killed by these CTLs. On the contrary, the quiescent neural stem cells evade this killing. So this happens pretty much as in the periphery. But there are a couple things here that was one thing that was surprising to us was, in contrary to the periphery, we couldn't kill all the cells. So we thought, well, maybe because the T-cell trafficking is not as, let's say, probable or frequent as in the periphery, maybe we have so many GFP-positive cells that the T-cells that access are not enough to kill them. So we decided to check that by focusing on the cortex, because of course we were using a GFAP-CRE mouse, so all astrocytes in the brain are green, are GFP-positive. And this is the result. So in the cortex, we also see infiltration of the T lymphocytes, and again, we see partial killing of those astrocytes. So to check if that was indeed a matter of the ratio between T lymphocytes and GFP-positive cells, we decided to do an experiment in which we could have fewer GFP-positive cells. And to do that, we decided to use electroporation. So at E15 in the mouse, one can electroporate in the lateral ventricles. In this case, what we did was to electroporate the GFAP in an integrative plasmid that is dependent on transposase, and transposase under the control of an astrocytic promoter in an episomal plasmid. That, what you at the end, what you have is GFP in GFAP or glass-positive cells in the niche. This procedure labels much fewer cells, so many fewer cells. And you can see here the cytometry, the proportion of cells that are being labeled in each subpopulation is very low, around 2%. In this case, we could see that the GDI T cells almost eliminated all the cells that had GFP, but again, if we looked specifically in the quiescent pool, they were not eliminated. So it could be that in the brain, T cells can eliminate cells within the brain, exhibiting neoantigens, but they are not trafficking in numbers high enough to eliminate many more. And that's a thought for neurodegeneration, is maybe in neurodegenerative diseases, maybe the number of cells that have abnormal proteins may be a limitation with regards to the capacity of the immune system to enter the brain. That's a thought. This is some speculation, okay? So we checked then MHC1 complex molecules at the membrane. And this was really surprising, because if one takes all the different populations in a naive animal, and checks for MHC1 proteins at the membrane, and this is the alpha chain subunit. You can check that by the alpha chain subunit or monitoring the presence of beta-2 microglobulin, which is the other subunit of the complex. But if you check, none of the cells exhibit any MHC1 complex at the membrane. And that also happens with neurons across the brain. That's the normal situation for all neurons in the brain. Here you see the only exception, which is microglia, which does express these complexes. But in the animals that were injected with the GIII cells that were susceptible to the killing, then you easily detect the MHC1 complex at the membrane. So that was not surprising, because as I told you, neurons and also the cells, as we can see, can respond to interferon gamma secreted by the T lymphocytes and then put MHC1 complex at their membranes. But what was intriguing was that all of them did so. He exhibited MHC1 complex, MHC1 at the membrane. Of course, the activated NSCs, which are the ones that are killed with more efficiency, have the highest levels. But why QNSCs were evading the immune attack if they also put MHC1 complex at the membrane? First, I'm going to show you that indeed, if we stimulate, for instance, neural stem cells which do not have MHC1, if we stimulate it with interferon gamma, they start putting MHC1 at their membrane. We can do that. We can check whether that interferon gamma is coming from GIII cells in an in vitro approach. We can take GIII cells that have been activated because they were injected in an animal, isolate them, put them in indirect co-culture with neural stem cells, and either use a control antibody or an antibody anti-interferon gamma. And what you can see is that in the presence of the GIII cells, the neural stem cells in the neurospheres put MHC complexes at their membrane. But if you block with anti-interferon gamma, you go back to the basal situation without any at the membrane. Again, let me go back to this puzzling situation. Quiescent NSCs did have MHC1 complex, and if you do so, if a cell does so, is susceptible to CTL killing or not. We have learned from cancer cells that the interaction between the T lymphocytes and a cell is dependent on MHC1 complexes and many modulatory proteins. Some of them are immune checkpoints that are being used now in immunotherapy because cancer cells put these immune checkpoints, these ligands for these receptors in lymphocytes at their membrane to calm down the attack of lymphocytes. And one of these is PD-L1, and for instance, we are working in mice that were generated by Oscar, for example. But what I show you today is that indeed, if in animals that are inoculated or they have GIII cells injected, what we see is that now we have an opposite pattern. Quiescent NSCs put at their membranes, have at their membranes, under these conditions, higher levels of PD-L1 than activated NSCs. So these cells are armored with tools, right? They have, quiescent NSCs have very lower levels of MHC1 complexes and higher levels of PD-L1 than activated ones. So this tells us that this is going to be a complex situation. But maybe we can, what happens if we manipulate a level of one of these molecules, right? And this is what we thought of doing. And we thought, well, maybe this is transcriptional. As in the periphery, Judith had shown that was transcriptional regulated. So we decided to check the same thing. And we looked at our transcriptomes, right, for our NSCs. And indeed, as in the periphery, this transactivator for the genes is downregulated in the quiescent cells. Well, that was like in the periphery. But look at this. This is the expression. Again, it's a transcriptome. This is the expression of the genes that encode the MHC1 complex subunits. And these are the genes that encode for the molecules that transport the peptides that are generated in the cytoplasm by the proteasome to the endoplasmic reticulum, so thereby they can bind to the MHC1 complex in their traffic to the membrane. So those are required also to assemble the whole thing. So when we saw that these genes were highly expressed, we thought maybe this is a post-transcriptional regulation or a regulation of the trafficking of the molecule, and this is the hypothesis that we thought about checking. And as I told you, this MHC1 complex traffics from the endoplasmic reticulum to the Golgi to the membrane as any membrane protein. And Pablo, what he did was to measure the total levels of MHC1 complexes and the extracellular levels differently. And what you see here is that, again, this is the alpha subunit of the complex. If you have basal condition, you have MHC1 proteins, but none of them are extracellularly exposed. But when you induce with interferon gamma, then now you have the MHC1 complex at the membrane. So it was a matter of trafficking. But it turns out that, and you can also check that, no, it turns out that there is little information, very little information about how the trafficking of these MHC1 complexes is regulated. There are only 3 poorly known molecules, and we decided to test one of them. And this is CD99, encodes for a transmembrane glycoprotein of unknown ligand. But we saw in our transcriptomes that CD99 was differentially expressed between activated and quiescent neural stem cells. So we checked at the level of the protein, at the membrane, because of course CD99 is a membrane protein, as I told you. And we could see that activated NSCs, acutely isolated from our niche, have higher levels of CD99 at the membrane. And you see here the data, the quantitation. And furthermore, Pablo was able to co-IP the alpha subunit of the MHC1 complex with CD99. So they are bound together in our cells. So I'm going to show you the data we have now. And this is an unfinished experiment because this is not only unpublished, but it's also unfinished because I wanted to tell you what we're doing and the ideas behind. So, so now we know that CD99 indeed regulates exposure of MHC1 in response to interferon gamma in vitro. And we can interfere with the expression of CD99 or overexpress cDNA for CD99, and in both cases, the levels of MHC1 at the membrane goes with the level of CD99. So we have also done the experiment with these changes in the expression to see that whether Jedi T cells can kill more easily cells in vitro, and that has worked. And what I cannot show you, because it's currently underway, is that we're using electroporation to modify the levels of CD99 in vivo to see whether in the adult, then the Jedi T cells are able to kill quiescent neural stem cells. So what I have shown you is that the neural stem cells, the quiescent neural stem cells also have mechanisms to evade being killed. In many adult niches, the quiescent stem cells are the reserve stem cells. They are the ones that sustain cell production over the lifetime. So these are precious cells. But they do not do it as in the periphery through transcriptional regulation. They have other layers of regulation, including this post-transcriptional regulation and and the regulation of trafficking. And also we now need to look into PDL1 and its regulation because it's going to be 0, 0, 0, 0. I'm done. Because we see that these cells have other mechanisms, and as I was telling you, they are armored to combat T cells. These other mechanisms, and we are also checking whether there could be even niche effects that could modulate these interactions. They are quite complex. Okay. This is the group. I have shown you especially the work of Jose Manuel Morante in the cytometry, and Antonio Jordan is our bioinformatician. And of course, this is the work. This is work of Paulo Martí and the funding agencies, and I thank you all for your attention. Thank you. Ya me voy bajando, ¿no?
Isabel? So that was very beautiful. I mean, this CD19 regulation, is it specific for neural stem cells? Is it the general phenomenon that regulates MHC class 1? I mean, I was wondering, for instance, in the case of resistance to immune therapies, that is often found to be linked to deletion of beta-2 microglobulin. And if this were to be a general phenomenon, you also expect to see differences in this molecule. You just wonder if you ever looked for that, because it seems an interesting approach.
Yeah, good question. I mean, there is not much known. For many years, MHC1 complex was, trafficking was bulk. So it was passive. As soon as it got produced, it went to the membrane. Some viruses can hijack that traffic, of course, so the T cells do not kill the cell. And there is one molecule that we don't know if it's implicated that was identified looking at viral defense. This CD99 has only been described in the immune system because, of course, T cells also have MHC complexes at their membranes, and it's regulated. As I was telling you, now we realize that this trafficking is not passive, it's regulated, but MHC1 complexes do not have peptide signals for the traffic, so you need a cargo molecule, and that's why we proposed that we needed a partner. CD99 has been described in some immune cells and nothing more. So we think that it's a new mechanism, and it would be nice to know whether it operates in neurons, for example, or in cancer. Yeah, but there is no more information. We were lucky enough to look for the one we thought, and it turned out to be differentially expressed. Carlos.
Very elegant, Isabel. So there are many, many questions that these results pose forward. So I wonder whether both parts of the niche, the one at the subventricular zone and the subclavicular zone, are equally attacked when you do these manipulations, and whether there is any, or you predict any consequence of those.
They should be. I don't know, because of course they're going to be attacked. I mean, and we can see that they attack astrocytes. In the hippocampus, the distinction between quiescent and activated cells are not that obvious or that established. One thing we could do, because we don't have this, we have done all we do the phenotyping by cytometry, so that gives us a high level of resolution. But in the hippocampus, what we're doing, just to generalize, although that's territory of Maria and other people, I mean, we don't work in the two niches, is to look by immunohistochemistry, because we can see the CD45 lymphocytes, we can see if they are active. If we see them close to cells that are proliferating, we can do that to extend to generalize. But we have been working more on the mechanism that may regulate this instead of, yeah. But yeah, we'll take a look.
Thank you very much for your talk. And it's kind of a two-part question. The first one, do you think this JEDI system could be used to kill other cells in the brain? So, for example, to create like a knockout mice, but just killing some cell lineage? And also, have you seen any side effects of these killer cells infiltrating the brain and start killing, like inflammation or something like that? Thank you.
Excellent questions. The first one, I think you're trying to say whether this could be used to ablate cells. I guess, yeah, it could be used to ablate. And then the second question is related to the first one, in case that the T cell infiltration is not producing other things, right? There have been papers showing and indicating that there are a few immune cells in the neurogenic niches that may condition with paracrine signals the cells. With RNA-seq analysis, it's very difficult to quantitate and monitor T-cell infiltration with immunocytochemistry, but it has been done with single-cell RNA-seq. And what they described was that you have T-cells of the CD8 positive and CD4 positive in the subependymal zone, that these cells increase in number with aging, and they can regulate the behavior of neural stem cells. I can tell you that we have done the experiments, our phenotyping, in RAG1 knockout mice, which do not produce any T cell at all, and we have found no difference whatsoever in the lineage. It could be that during aging, they have, because there is also an increase in interferon gamma in the niche. I cannot discard that during aging. And during aging also you have a blood-brain barrier that's more permeable, as it could happen in a neurodegenerative disease. So this is a very basic situation that may inspire, and that's why I decided to talk about this, may inspire, I don't know, some concepts or thoughts in neurodegenerative diseases. Sonia. Yeah, the same. I mean, yeah. Correct. That's my expectation, except the quiescent ones. But I can tell you one bit of information. When Judith published the paper on the tool, on the Jedi mouse, she did an experiment using the Jedi T cells in a microglial reporter mouse, the CX3 CR1 GFP mouse. And she was able to kill all the cells. But there is, and related to what you were saying, the difference was because you have that reporter is also active outside in the periphery, because monocytes also express it. She had to do an irradiation to ablate the periphery and transplant non-reporter monocyte, hematopoietic stem cells. So she irradiated. And we all know that irradiation increases neuroinflammation and disrupts blood-brain barrier. And she killed all the microglial cells in the brain. So my expectation is that if you do a stroke, increase neuroinflammation or whatsoever, you're going to kill all the GFP cells in the brain. And that could happen also again during aging or in neurodegeneration.
Here, it's a very fast one. It was just an amazing talk. I'm really happy to listen to you. You are super inspiring, and really for young people to continue to research, because there is always something new to continue working on. So my question is, do you think these results can be translated into other cell types like quiescent muscle stem cells and so on? And do you think with age, even though this population is decreasing in numbers, also can increase the expression of these antigens, these MHC1?
It could be very nice to see it in muscle, because they are quiescent for the entire life. And it could be very nice to see it when you injure, when they become activated, and see whether it's, again, a transcriptional mechanism as in the hair follicles or not. But that's an experiment for you.
Thank you.